This is the fourth video under unit 2 of the subject evolution. We were talking about Oparin and Haldane's theory, and we looked into the first three stages of chemical evolution according to Oparin and Haldane. So the fourth stage is the formation of colloidal system. Here, the organic molecules that were produced in stage 3, they got dispersed in a medium of water to form a colloidal system. Now, a special property of colloidal system is it helps molecules come together. So, protein molecules started coming together. They started absorbing and reflecting light, and they couldn't move around. They carried negative and positive charges. They attracted each other or repelled each other. And this was the first step in the transition from a non-living to a living system. That is, the particles of the colloidal system started coalescing, coming together and forming a coagulum. They were then distinctly separate from the medium. That is, they started forming a membrane around them. And this is the next stage where this membrane covered droplet which got separated in this colloidal medium is now called a coacervate. And the process of formation of coacervate is called coacervation. These are considered to be the primary living molecules. These were relatively unstable and they were easily affected by changes in pH, electrolyte, pressure, electric field, etc. However, they had an ability to absorb and store substances. And in time, they started utilizing energy and they formed complex coacervate systems. In the next stage, these coacervates, they acquired the capacity to divide and enlarge. So when any substance starts dividing, we call it a living organism. So these are then called the protocells or protobionts. Inside these, enzymes started forming, internal reactions began taking place and led to the formation of living molecular systems. So they had properties like mutability, self-duplication, heterocatalysis, that is, they had the property of life. This picture shows how, due to lightning and ultraviolet radiations in the primitive atmosphere, inorganic compounds join to form small micromolecules. These micromolecules started joining to form complex macromolecules. Macromolecules then started forming coacervates. These coacervates then in a colloidal system separated from each other started forming the first living cell capable of mutation, capable of duplication and capable of reactions. Now Oparin and Haldane's theory was only a hypothesis when they proposed it. That is, no one was able to prove whether Oparin and Haldane's theory is possible. But in 1953, it was Harold Urey and Stanley Miller who gave the first experimental evidence for Oparin and Haldane's theory of chemical evolution. They had an experimental setup and they were able to synthesize simple organic compounds from inorganic compounds. Let us look at how their experiment was conducted. They took a high energy gas chamber, they filled it with water in the form of water vapor, ammonia, methane and hydrogen. They had a boiling flask which was boiling water in order to produce steam. There was a condenser for cooling the contents that were being produced. And there was a liquid chamber to collect this condensed liquid. And there were many connecting tubes for the circulation of the gaseous contents. So can you look? There is a big chamber where there is water vapor, methane, ammonia and hydrogen. Energy was supplied in the form of an electrical spark water in the form of heat was circulated in this setup, then whatever was being produced was cooled and then collected. And to their surprise, they found that there was actually amino acids and other organic molecules found in the condensed liquid. They kept this experiment for 1 week and collected the liquid in the liquid chamber. They analyzed the chemical composition and they found that there were several amino acids such as glycine, alanine, aspartic acid, glutamic acid, etc. There were fatty acids, simple sugars, adenine and urea. You should remember that in the beginning of the experiment, Urey and Miller did not supply any kind of organic molecule in this experiment setup. But at the end of the experiment, they were able to synthesize amino acids and other organic molecules, thus proving that it was possible for Oparin and Haldane's theory to have happened in the beginning or the origin of life on Earth. Several other scientists also tried to prove Oparin and Haldane's hypothesis. In 1954, Bahadur produced a broad variety of amino acids when they subjected a mixture of paraformaldehyde, ammonia and ferric chloride to the action of strong light. Two more scientists, Pavlovsky and Pasinsky, they synthesized amino acids such as alanine, glycine, aspartic acid and glutamic acid when they exposed a mixture of gases first to electric spark and then to UV radiation in the year 1959. Sidney Fox, when he heated a mixture of 18 amino acids at a very high temperature of 200 degrees in the year 1964, he was able to produce stable protein-like molecules called proteinoids. And when these were cooled, they formed spherical cell-like aggregations which he called microspheres. In 1963, Ponnamperumana and Sargent and Marina, they produced adenine monophosphate, adenine diphosphate, adenine triphosphate, adenine guanine and adenosine by thermal polymerization, that is using heat. Lowe and others could synthesize adenine, guanine and hypoxanthine. So not just Urey and Miller, several other scientists were able to prove that organic molecules can be produced from inorganic molecules. That is the end of the theory of biochemical evolution by Oparin and Haldane. We look at the unit in the next